Our next speaker is Dr. Adam Durbin. He works in the Division of Molecular Oncology at St. Jude Children's Hospital. He received his BS in biology at York University in Toronto and his PhD and MD at the University of Toronto. He completed his residency at Boston Children's Hospital and his fellowship at Dana-Farber. His research includes epigenetic and transcriptional regulation of high-risk pediatric cancers, including neuroblastoma, chemical biology, and drug discovery for pediatric high-risk tumors. He has received numerous awards for his research, as well as all of our presenters today, and is the author and co-author of numerous publications. And his talk today is developing new preclinical tools and approaches to targeting neuroblastoma. Dr. Durbin.
Thanks, Pat. I'm embarrassed by all of that. So I think what I want to talk to you guys about today is a couple of things that we're doing in the lab, and not just in my lab, but as a community, with sort of most of the neuroblastoma community, to try to understand what makes neuroblastoma cells tick and how we can take advantage of that to develop new therapies. I have a lot of disclosures. I've received funding from a number of agencies that I think folks in the audience here are probably very familiar with, including the NIH. I'm a St. Jude employee. That's where my lab is. That's where my clinical practice is. So I receive funding from St. Jude. And I want to point out, as a necessary piece of doing drug development, we have to patent things. Otherwise, pharmaceutical companies have zero interest in pursuing these things. And so we have a lot of patents. And I'll point out, to give you a sense of how sort of hot off the presses the stuff that I'm going to be talking about today is, one of the patents was filed last week. So this is like as up to date as we can possibly go. So Pat mentioned most of my path. I'm just going to say a couple of words about it. I started, I'm Canadian. I started at this university called York University, and initially was going to become a cardiologist. And you see how that turned out. And so after finishing my undergrad degree, I enrolled at University of Toronto, where I met this guy. This is David Malkin, who is a pediatric oncologist, works on hereditary cancer predisposition syndromes. And in David's lab, I studied pediatric sarcomas, worked mostly on rhabdomyosarcoma and osteosarcoma, where I got my PhD. And then at University of Toronto also got my MD. Before moving on to Boston Children's and Boston Medical Center, where I did my residency in general pediatrics, progressively getting farther and farther away from this idea of becoming a cardiologist. While I was at Children's, I did a rotation in the ICU. And as a very young doctor, ran across a patient who was extremely ill with high-risk neuroblastoma. And that completely changed the course of my clinical practice and my research practice, everything that I would do subsequently. I went out and actually found one of the local experts in neuroblastoma, who is this guy, who is downstairs right now. His name's Tom Look. He is a sort of visionary in the world of neuroblastoma basic science research. He's one of the people in his lab. ALK was discovered as being mutated in pediatric neuroblastoma. And so I worked in Tom's lab for a total off and on during training of almost 10 years. So you can tell I started when I was 3 years old, right? So, okay, so I then went off and did a postdoc and clinical fellowship training at Boston Children's and Dana-Farber, where I sought out this person. This is Kim Stegmeyer, who's another sort of a very senior investigator in the world of pediatric cancers, and in particular neuroblastoma, where I got involved in a project that I'm going to tell you about today called DEPMAP, which is, I think, foundational to our study of neuroblastoma. And then ultimately, after attending on service and continuing to be sort of a instructor at Dana-Farber and Boston Children's, I ultimately, in 2020, the perfect time to do anything, not like there was a pandemic going on or anything, but in 2020, I left Boston and moved to St. Jude, where I opened my independent lab. And that was really led by this last guy, Charlie Roberts, who is an exceptional scientist, clinician, and whose lab focuses on studying a different pediatric solid tumor called rhabdoid tumor. Okay, so that's who I am, or at least where I trained, and this is what we do. And so before I go through the boring of what we do, I want to point out this patient here. This is great because for two reasons. So first, it avoids any HIPAA issues, right? Because trust me, there's no way any of you know who that is. But this young lady has high-risk neuroblastoma. This is one of my patients. While she seems to like me, when I come into the room, she hates chemotherapy, and so she hides in the curtain. She's not that hard to find, but in any case. And so what we do in my lab is try to use chemical biology and functional genomics to study neuroblastoma cells themselves to figure out ways to both identify potential targets, and you've heard a lot over the last 2 days about this is a target and this is a target. More than just identifying the targets, we want to understand why they're a target, and we want to develop new drugs to target the target. That's what we do. And our goal is to try to reduce toxicity of current therapies by being more on target, hitting those things that the cancer actually needs, and improving the efficacy of treatments that we already have. And one of the most important things that I can say about our work is that we are entirely open access. So everything we do, we give away. We make new compounds, we give them to anybody who asks. We generate data, we make it publicly available to everybody. And I'll show you a couple of examples of that. And that's because we are only so good, and the world is full of amazing scientists who want to take what we do and build on top of it. Okay, so my disclaimer is what I'm going to tell you about today is not ready for clinical trials. So I'm going to ask for the impossible, which is understanding and time in a disease where I know we have no time. Okay, so I'm going to tell you two stories. One's going to be a little longer, one's going to be a little shorter. And the first one I'm going to tell you about is how we've started to use new methods to find new ways to try to target neuroblastoma cells. So Angelica actually showed this slide, but didn't spend a lot of time describing it. And this is a very famous slide. I don't know if folks have seen this before, but this is a very famous slide in the world of cancer biology. So in 2018, a group from St. Jude and then several other groups around the world actually published very similar data that looks like this. And what you're seeing here is, let's see if I can, yeah, okay. So what you're seeing here is the number of mutations in the genomes of all these different types of cancer. And along the x-axis here are lots of different types of cancer. And neuroblastoma is in this sort of like light blue color. And it's separated. On this side is all the pediatric diseases, and on this side is a bunch of adult cancers. And what this slide is meant to show is that adult cancers have lots of mutations, and pediatric cancers don't. And so what that means, practically speaking, is that when we examine adult cancers, oftentimes there are mutations, and that tells you where to go to build a drug, because you have a mutated protein, and that mutated protein typically has some activity that can be disrupted. It has some new activity that's driving the cancer, and so we can try to develop a drug to target that. But pediatric cancers don't have a lot of mutations, and it turns out that the mutations that they do have are oftentimes in things that are really hard to target. So we're a bit stuck in terms of how do we develop an understanding of what the best next target is to go after. So I think about neuroblastoma much like I think about many other pediatric cancers like this car. So I'm not an auto mechanic. I have a Mini Cooper. It breaks down all the time. And what I have learned is, first, don't buy used cars from the guy who sold it to me, but also there are a lot of ways you can break a car. Because my mechanic tells me that there's some new thing in the car that's broken this time, and that's different than the last time. When I think about neuroblastoma, I think about a couple of genes that I think are very familiar to folks in the audience. Things like ALK, MYC, MYC-N, ATRX in some cases. These are things that we know are mutated in the disease and drive neuroblastoma. To me, these are the engine of the car. They're the thing that make it go. But it's almost never the engine that breaks in my Mini. It's usually something else. If the roads are really bad, I might blow a tire, you might lose a fan belt. I don't know anything about cars. You can tell I'm limited to fan belt and tires. But basically, you can imagine you can stop this car by hitting a lot of different areas on the car. You could, lose a tire and then the car is not going to go, or you could cause some other damage to the engine, or some other part of the car that would stop it from going that isn't targeting the engine itself. So that's exactly what we have taken the tactic of doing, and that is because many of these genes, sure, we have drugs like lorlatinib and crizotinib that target ALK, but we don't have drugs, at least not right now, that target MYC and MYC-N. And there are large groups that are working on trying to build those compounds, but these have been a major challenge in cancer biology for decades. And so in the interim, we need something else. And so how can we figure out what are the right targets to go after? All right, grade 11 biology. We're going back to the central dogma of biology. All of the information that is coded that tells a cell what kind of cell to be is found within your DNA, and that's organized into genes, which make up about 1% of your DNA. The rest of the 99% of your DNA are all made up in elements that tell you which genes to turn on when. And so you can imagine if every cell has the same genes, what makes a heart cell and a skin cell are which of those genes are turned on and off. When those genes are turned on, it makes this intermediate called mRNA, which looks sort of like DNA, but then a system called the translational system comes in and turns that into protein. And it's the cohort of proteins that are found within a cell that tell that cell what kind of cell to be. So how can things go wrong in cancer? In some cases, you can imagine you have the damaged instructions. So let's say you make thousands of copies of the MYC-N gene. That leads to too much intermediate, too much effector protein, and can lead to the development of cancer, in this case, neuroblastoma. So why am I telling you about this? Because over the last 20 years, there's been a number of technological advances that folks may have heard of because they've been covered a lot in the media. And one of those is this thing called CRISPR. So CRISPR is a basic tool that gets used. It's built out of, it's actually based on a bacterial defense mechanism. And essentially, you can think of CRISPR as being a way to damage any gene you want. And so this allows us to systematically damage any of the 22,000 or so genes that are found within any cell. And so you can imagine a lot of very miserable scientists sitting around going one by one, trying to damage each of the 22,000 genes in one cell to figure out which are the genes that are actually important to make that cell grow. But you can imagine if you did that, some of those genes, if you damage them, might actually cause these cells, let's say these are neuroblastoma cells, to grow faster. Others might have no effect at all, and some might actually cause these neuroblastoma cells to die. And those are the ones that we want to know. So essentially, over the last 10 years or so, there has been a very large-scale project to do this exact technique across every gene that exists in the genome in hundreds of cancer cells. Different types of cancer, including neuroblastoma, to ask the question, what are the genes that are important in each of these different types of cancer, and how can we now go after those genes in particular? And so that is this sort of philosophy here, where we take cancer cells that are derived from different patients' primary tumors. We take them and we study their basic DNA and their RNA and protein complement. We also take them through these knockout screens where we've damaged every gene in the genome. We also expose them to different types of drugs, including chemotherapies, as well as sort of novel therapies. And then we integrate all of that to try to drive some therapeutic hypothesis. And I'll point out that I've got a website listed here, depmap.org. That's because everything we do as a part of this project is posted online. And it is all freely accessible to any scientist in the world. And as a result, over the last 10 years, there have been thousands of papers that have been generated and clinical trials that have been started out of this data set. In 2021, we published the first map of pediatric cancer dependencies. That's what we call these genes that are important in different types of cancer. And you can see here the distribution. In total, we did 84 different cell lines derived from different patients' primary tumors. This is heavily overrepresented. Heavily overrepresented for neuroblastoma, and that overrepresentation has continued. And so currently, we've tested somewhere on the order of 30 or 35 different neuroblastoma cell lines, and we're continuing to expand that collection. This has grown to a level that we had to come up with a new name for it, and so we called it the Pediatric Dependencies Accelerator. This now includes more than 100 investigators nationally as well as now internationally. And this has been focused on specifically pediatric cancers, and one of the hallmark tumors that we are studying is neuroblastoma. My lab happens to be one of the labs that is leading the initiative in neuroblastoma itself. This work is all being done at Dana-Farber, Boston Children's Hospital, St. Jude, and the Broad Institute. Although this has international reach, we are bringing in models from around the globe. We are sharing data with other centers. We are specifically doing our best to sort of house the data in a way that is accessible and easy to use for people. As I mentioned, hundreds, now thousands of papers, scientific programs. It's entirely open access, and it's community responsive. And so I gave a talk about what we were doing in neuroblastoma to the Neuroblastoma Drug Development Symposium in Boston in 2024, not that long ago. And the community, the neuroblastoma community told me, hey, what we need to do is prioritize specific subtypes of neuroblastoma. We need to know more about those subtypes that have problems with telomere maintenance that you heard of from Angelica. ATRX, and this sort of new recognition of a subtype of neuroblastoma called mesenchymal neuroblastoma. And so we did that. And in the last 9 months since that meeting, maybe closer to a year, we have now put into the pipeline another 15 models that cell lines derived from patients that reflect those different subtypes so that we can understand those subtypes even more with a goal, again, of deriving new targets for those patients. So as I said, we've increased our focus on rare subtypes. It turns out cells in a dish that have been cultured by many different researchers over the last 30, 40 years, those kind of change, and they might not look like the cells that are in the patient's primary tumor. And so we've now been working with a group called the Cancer Cell Line Factory so that biopsies and resection samples from surgeries go straight to this group that makes new cell lines so that they are as close to the patient as possible, and those are the cell lines that we're now prioritizing for screening, trying to get as close to the patient as we possibly can so that we can study those cells so that we can try to make a difference. We're integrating more human tumor genetics, predictive biomarkers. We also recognize that what we're doing here is destroying genes. We can't do that in people. What we can do is deliver drugs, and so we are using new types of chemistry that I'll show you that do sort of a similar thing. They destroy the protein instead of the gene, but they sort of have the same effect. And this has led to lots of clinical trials. So what have we learned? So in 2018, when I was a postdoc, we published the first map of those genes that we thought at the time were selectively required by neuroblastoma cells. Genes that seemed to be highly specific for neuroblastoma and only needed in neuroblastoma cells. And we got a list of 150 genes. And the community has grabbed onto these genes, and everybody has been studying them. Many of them nobody had ever heard of before. We had no idea what they did or whether we could target them in any way. Some of them made sense. We saw MYC-N, we saw ALK, we saw ATRX. So these things made sense. And as the number of cell lines went into DepMap, or into the Pediatric Dependency Accelerator, we saw the same thing over and over and over. Some of the genes shuffled a little bit, and we found new targets that were more specific. But what we kept seeing was this process of transcription, how genes are turned on and off. That was really important in neuroblastoma. And so this is a figure that we published in 2018. Each of these individual bubbles is a gene that is individually required for neuroblastoma cells to grow. And so if you lose any one of those genes, the cells will stop growing. And you can see MYC-N is here, so that makes sense. And anything in green is a transcription factor, and MYC-N is a transcription factor. It's a gene that turns on other genes. And we've taken this and tried to study this to figure out how these proteins actually interact with each other and how they work. And what we found was that they establish a circuit, and that each of them drives each other. And now, instead of just studying MYC-N, we have all of these other, so I think of them as buddies of MYC-N. And if you destroy any one of these, you lose them all. And so we actually don't need to target MYC-N. That would be the best target, in my opinion, for this. But if we targeted this gene, ISL1, we would have the same effect as targeting MYC-N. So this is great. This is more shots on goal. And so we've thought for the last 8 years, how can we try to build new drugs to target this circuitry? And we have done a lot of work trying to do that using different technologies, and I'm going to tell you about one of them. But it's not just been us. In fact, the community has grabbed onto these different genes and is really trying to study them, including most recently, John Maris's group published a new CAR T cell that targets this one, FOX2B. So, and that's in early phase preclinical development now. So one of the things that we noticed when we were studying this circuit is that every one of these genes was regulated by something called a super enhancer. And this is found in the 99% of your DNA that tells your genes which ones to turn on and which ones to turn off. And it turns out, not just each of these being regulated by a super enhancer, but it turns out that sitting right in the middle and the most interlinked protein out of all of these proteins that was important for neuroblastoma was this gene called P300. And P300 is a gene that makes super enhancers. So this totally made sense. It was like all of these genes that are important are really controlled by this guy, P300. And so for the last 8 years, we've been studying P300. And this is the structure of P300. It's a humongous protein. It has a brother called CBP. And what these two proteins do, they're found in every cell in your body, both typically at about the same levels of expression. And what these proteins do is they make these enhancers that tell your cell which genes to turn on and which genes to turn off. And we think that P300 and CBP play very similar roles in normal cells. We think you can use either one of them. But it turns out in cancer, many cancer cells pick one or the other to rely on. And about 1/3 of all human cancer requires P300. And we know that from our genetic data. Initially, we thought this was really specific for neuroblastoma. Subsequently, we actually learned that it is specific for neuroblastoma, but it's also specific for several other pediatric tumors, including rhabdomyosarcoma, Ewing sarcoma, osteosarcoma, and brain tumors like medulloblastoma. And so what you're seeing here in red, this is neuroblastoma, and the higher you go, the more specific you are. And so here what you can see is that neuroblastoma is pretty specific for P300, and it doesn't seem to need CBP at all. And I'll tell you, they're both there. When we look in the cells, they're both present, but they only need P300. And so this gave us an opportunity, and that was, could we figure out a way to target P300 and leave CBP alone? And we thought that if we did that, if we targeted P300, it would kill neuroblastoma cells, and we might not have so much toxicity, because in normal cells, they would still have CBP, and that could do the function of both of these proteins. So that was a very pie-in-the-sky idea, because 30 years of chemistry, people had only ever been able to develop small molecules that targeted both, because these two proteins, they're brothers, they look very, very similar. And so getting a molecule that can only bind to one of them has been an exceptional challenge. And so we used a new type of chemistry called degrader chemistry. And essentially, what happens is on one side, our small molecule binds to this complex, and on the other side, it binds to this protein, which is P300. And the binding of P300 to this complex causes P300 to get tagged, and then the cellular, basically garbage compactor comes in and eats P300. So what we are doing is teaching neuroblastoma with this small molecule to kill a gene that it needs for survival. Okay, we call this molecule J-quad1. This is the structure down here, and I think of it as chemical deletion. And when we do this in cells, what we see here, this is done in a MICA-amplified neuroblastoma cell line. We see loss of P300 with no effect on CBP. Neuroblastoma cells do not like this compound at all. What we see when we treat with this compound J-quad is cleavage of caspase-3 and cleavage of PARP. These are marks that the cell is killing itself. And we can take this small molecule, this is a first generation small molecule, a tool that we made in the lab. This is not a drug yet, but we can take this and put it into mice that have neuroblastoma. And when we do that, we see the tumor is slowing down. And when we look at how they're feeling, we literally do liver function tests, kidney function tests, blood counts. We look for any evidence of toxicity in the animals. There is none. And when we look at all of those, we do all the same things that we do, checking counts, that sort of stuff that we would do clinically. So we're pretty happy with this idea that we can slow down the tumor with very, very limited toxicity. So now we've been asking a lot of questions using this molecule. What are the effects at a basic science level? Is there resistance that will evolve to this small molecule? We see the tumor slowing down. We don't see the tumor being cured. And so we've asked, can we combine it with other therapies? Can we combine it with conventional therapies, chemotherapy, immunotherapies, to try to get better effects? And does it matter how you target these proteins? Because there are lots of different small molecules that will do that. And in fact, yes, it matters a lot. And so what we are doing right now is taking this tool with the help of the community and several other agencies, and really being led by St. Jude, to try to develop this into a drug, improve its activity, so that hopefully we're not just slowing down tumor growth, but we're actually seeing cures. Okay, in the last part, I want to talk to you guys a little bit about cell state and new ways of thinking about neuroblastoma, and specifically what we're trying to do to address the problem of relapse. So for the last 5 years, folks in my lab have been very focused on thinking about how neuroblastoma cells change when the disease comes back. And we know a bunch of things. Historically, from genetics and from other studies, we know that neuroblastoma cells will actually increase the levels of drug efflux pumps. So we give doxorubicin, for example, and it never makes it into the neuroblastoma cell. That's not entirely true clinically, but these cells, when receiving chemotherapy, have the ability to pump it back out. And that's a problem. In addition, these cells will undergo genetic mutations, and then they have this really mouthful of a phrase called non-mutational epigenetic reprogramming of transcription. And people have really studied the first two and this third one, and which, again, horrible name, is something that people don't know a lot about. And what this is this concept that as cells are exposed to different stimuli, chemotherapy, low oxygen, nutrient deficiencies, that they change. And they change in a way that's flexible. And the basic idea here is that different genes turn on and off, and that causes changes in cellular properties. And this is exactly what we've been studying. This is all the way along the same sort of principles. And we have known for many, many years that neuroblastoma cells do this. So here are some neuroblastoma cells that are growing in a dish. And I know that if I take this dish of cells and I treat them with retinoic acid, which many of you are familiar with as a sort of core therapy as a part of maintenance in neuroblastoma, that they will change, and profoundly. These cells essentially look like neurons. And that's the reason why we give patients retinoic acid, because we can drive this change. But this is not caused by mutations in the DNA. This is caused by the treatment itself. And so looking back through history, we've actually known that there are these different types of cells that are found within neuroblastoma tumors since the 1970s. And so here, for example, are two different cells. You can see they look pretty different. These came from the same patient's neuroblastoma tumor. So there are different cells that exist within the neuroblastoma tumor. And it is only in the last 10 years or so that technology and sequencing analysis has allowed us to figure out by these different methods that these cells have different properties. And it turns out that by using multiple different sequencing technologies, we can find cells that exist in one state or another. So I'm going to tell you about two cell types. One of them is called the adrenergic cell. This is the majority of cells within the neuroblastoma tumor. These cells are relatively sensitive to chemotherapy. They're relatively sensitive to most of our targeted therapies. And we know a lot about these cells because we have lots and lots of cell line models and PDXs and other sorts of ways of studying these cells. There is a much rarer cell called the mesenchymal cell. This is a rare and relatively chemo-resistant cell. We think it's resistant to many of our targeted therapies, including ALK therapies and GD2 therapies like dinutuximab. And this cell, we think, may be responsible for relapse. And we have almost no models to study it. And so if we can't study it, we can't figure out what it's vulnerable to. And if we don't know what it's vulnerable to, we can't figure out how to fix this cell. And so folks in my lab really took up the challenge of trying to figure out new ways to identify these cells so that we can study them, so that we can figure out how to kill them. And so we took advantage of a lot of data that had been published and made publicly available, and we integrated hundreds of data sets. And we did this with the help of two very talented computational biologists at St. Jude, Brian Abraham and Yang Zhang, who literally looked for the needle in the haystack. You should have heard the curse words that they said when I asked them to do this project. After looking at more than 160,000 sites across the genome in hundreds of neuroblastomas, we found a very small collection of sites that we could use in order to mark these mesenchymal cells. So we took these different sites, and again, this is all very experimental biology here. We created a new type of virus that we could put into neuroblastoma cells. And when these cells are sitting in this mesenchymal state, they turn red. And so we can now just look under the microscope and see red cells. We can graft these into mice to form tumors, and we can biopsy the mice after treatment in order to look for these red cells to see what treatments cause these red cells to increase in number, which ones actually make them go away. We call it Trex, which stands for Transcriptional Reporter Element of Cell State. And here's an example of what some of these cells look like. So we have Here up at the top, we have this cell line SKNB2. This is neuroblastoma cells that are derived from one patient. And you can see almost every one of these cells is green. There's only one little red guy up over here. So this cell line is almost entirely adrenergic cells. It's very sensitive to chemotherapy. In contrast, we've got this cell line down here, which is called G-men. And essentially every cell within G-men cells is red, and G-men is much more resistant to chemotherapy. I'm going to sneeze. Or not. Or not. Okay. It turns out that when we put this system into most neuroblastoma cell lines, it doesn't look like that. It doesn't look like one or the other. It looks like something in between. And now what we can see is that in these different cell lines, this one, and many others, we can see some cells that are red, indicating that they're more mesenchymal-like and they're more chemo-resistant, and some cells that are not. And so we've shown now, and I can say again, this is like as sort of up to date as possible. This paper was submitted 3 weeks ago. And actually there's a presentation at ANR downstairs on Tuesday, which will be the first, actually you guys are the first public disclosure of this. So you guys can go tell everybody at ANR. But we're gonna tell everybody on Tuesday. So these cells have all the properties of mesenchymal cells. And we're now using these with the community to ask multiple questions. The first is now we can separate these cells. We can study these chemo-resistant cells and figure out why they're chemo-resistant and how we can kill them. So we can identify their vulnerabilities. We can try to reprogram them, and we've had some success at doing that. Taking these cells and giving therapies single doses of compounds that we think have very low toxicity that are in clinical trials now, to trick these cells into being more sensitive to our conventional therapies. Within studying their vulnerabilities, we want to study their genetics. We want to study using these as biomarkers in their active clinical trials, using these the signatures that we've used in order to find these cells in patients with neuroblastoma, and ultimately with a goal of getting new therapies. And I'll say the paper was submitted 3 weeks ago. The methods and cells that we've generated are already in 5 labs. And again, open access. We make everything publicly available. Everybody, 4 weeks ago, everybody who we have talked with about this system has asked for access to the cells. We have packaged them up and sent them off. And we're putting all of the data, everything that we've generated, into public repositories. So again, these are the goals of my lab. Again, reducing toxicity, improving efficacy, and we're all open access. This is my acknowledgement slides. I highlighted anybody who is a part of the neuroblastoma community. And so what I want to point out is this is not just me or not just folks within my lab. This is a group effort that is trying to study these cells, trying to study this disease in a way to develop new therapies. And of course, I need to acknowledge all of our funding sources. We receive funding from a lot of different people and different institutions or different foundations in order to fund this work. And I want to say thank you to you guys because you put your faith in us that we're going to try to make a difference, and that's what we try to do. This is my lab, and this is our mascot, which is a porch goose that I don't understand, but. And I want to say, everybody who's got an arrow is here at ANR. So I would encourage you, if you see them, because they're all walking around here, to go up to them, talk with them, ask them questions, and they're all happy to answer questions and be as helpful as they can be. And that's all I have to say. So. Hopefully I didn't go too over, and I think I'm right up against lunch, so. I don't know if anybody has any questions.
I heard the applause.
It was just polite applause. Everybody hated it, so.
Anybody have questions? See, I'm good at this part. Anyone have any questions? Or way? Nope. Go ahead.
I actually don't think I'm smart enough to come up with a question, but I am smart enough to recognize the work that you're doing and the value that it has, and to say thank you.
Yeah.
Thank you.
Thank you. Another one?
So I actually did have a question, as you have done very deep research into what seems to make neuroblastoma cells tick. Do we have any idea as to what seems to cause these cells to go dormant for what can seem like years at a time, like having a relapse after 5 years? Is that due to like the S style cells that causes relapse, but then they're responsive to chemo and antibodies, for instance, at first, and then maybe become resistant later down the road?
Yeah, it's a really good question, and this is an open and very active area of debate within the field. So much so that I think there's 3 sessions at A&R that basically are trying to address that question. What we think is likely to happen is that we think that our chemotherapies and most of our treatments work really well against these adrenergic cells. And they still work against these mesenchymal cells, but maybe not quite as well. We think that in human tumors, we think that these mesenchymal cells are the quiescent cells. And so what we think we're doing is we're treating away most of the adrenergic cells, and these mesenchymal cells are left behind. And given long enough, sometimes these mesenchymal cells wake up. And we don't know what causes them to wake up, in part because we didn't have any tools to study them. And so once they wake up, we think that these mesenchymal cells actually change back into being adrenergic cells. And so there's a lot of interest in trying to understand both what makes these cells wake up and what causes them to switch back and forth between these 2 different cell states. And once they switch back to being adrenergic, we think that they can grow very quickly and repopulate. And that causes, we think, a relapse. There's a lot of hand waving here and a lot of, we think this might happen. And that's because it's a really new area of study and has been really limited because of the fact that we haven't had the right tools in order to study it. And we hope that the tools that we've generated will allow us to ask those questions in order to figure out what the best way to target these are. So, but yeah, it's a really good question.
Anyone else? No? Okay. Thank you, Dr. Durbin.
Thanks, guys.